Welcome to this lecture on Mendelian genetics. We're first going to begin with Gregor Mendel. He is known as the father of genetics. He is the one that discovered the basics of heredity. So we'll go through that through the presentation, and then we'll also talk about Mendel's 3 laws. This pretty much applies to all organisms. So Mendel worked with pea plants, and the reason he used pea plants is because they're a model organism. They produce a lot of offspring, and they have a very short generation time. They also have different characters and different traits for each of those characters. So a character is such as, for the pea plant, like flower color. And then different traits of that character are such as a purple flower or a white flower. I know that's not white, but trust me, it's supposed to be white flower. Another example would be like seed color. We have yellow seed with a green seed. So those are different traits. So you can apply this to yourself. You have eye color. You have different traits of eye color. So you have brown eyes. You have blue eyes. You have hazel eyes. Those are just different traits of eye color. So we're just going to follow this experiment that Mendel went upon. Specifically, we're going to follow flower color. So here we have Mendel's experiment. So let me go through each of these vocabulary terms. So you have what's called true breeding. True breeding is where a plant produces offspring of the same variety. So if I take a white flower and I let it self-pollinate and it produces a white flower, that is a true breeding plant. But if I take a white flower and it produces a purple flower, that is not a true breeding plant. It has to produce the same flower color or the same seed color. So that's what Mendel did, is he made sure that he had 2 true breeding plants. He made sure he had a purple and a white. Both of these bred true every single time for that color. What he ended up doing was cross-pollinating these 2. And this created what's known as a hybridization, or a hybrid. So this in turn created this hybrid down here of purple flowers. Now he did this through the parent generation, which is the first generation. Then those flowers make the F1 generation. That flower made the F2 generation, and you can go on and on. So you can go to the F3 generation and such forth. So now let's talk about the law of segregation. Before we get into the law of segregation, we need to talk about certain vocabulary. We have gene and alleles. So a gene is the unit heredity of information of DNA. So this would be, an example would be like your hair color. And then you have different alleles of hair color. So you have such as brown, you have blue, you have red hair also. So those are just different alleles. If you go back in this video, a gene is a character, an allele is a trait. So dominant allele and recessive allele are written with letters. So a dominant allele is written with a capital letter, while recessive allele is written with a lowercase letter. And that's what the law of segregation is trying to do, is to separate these 2 alleles. So in this case, we had purple flowers. And then it separated each of those alleles so that each cell has a purple flower. That's the law of segregation. The law of dominance just states that a dominant allele will be expressed above the recessive allele. So some alleles are dominant while others are recessive. And that's what this is talking about down here. So next we're going to talk about Punnett squares and how Mendel used them to help determine his crosses. So first let's begin with the vocabulary. So phenotype is the physical characteristics. So in this case, the physical characteristic of these two flowers is purple and white. That's what we can physically see. A human example would be such as brown hair or red hair. That's what you can physically see. Then you have the genotype. What makes up those physical characteristics? And that's the genetics. That's the DNA. Now, genotype can be expressed in two ways. You can express it in homozygous or heterozygous. So there's two types of homozygous. So remember, those are the alleles. That is the letter. So if I write two capital A's, that is considered homozygous. If I write two lowercase a's, that is considered homozygous. So there are two ways that you can express a homozygous. A heterozygous is between different alleles. So this would be an uppercase and a lowercase. So these are two different alleles. They always come in pairs. Allele never does not have a pair. So if we go back to Mendel's experiment, he bred a purple flower, homozygous purple flower, with a homozygous white flower. And we put the purple flower here, we put the white flower here. And then we fill in the corresponding Punnett square, and this in turn gave us all purple flowers. So this is shown in his F1 generation. They all have purple flowers. So if we move on to the next F1 generation and allow them to, if we move on to the next F1 generation, allow them to self-pollinate. So when you self-pollinate, this is how you would make the cross. So I just correspondingly fill in the Punnett square, and then so we're going to have a purple flower here. We're going to have a purple flower here. We're also going to have a purple flower here, and then this one's going to be our white flower. So we come out with a 3-to-1 ratio of purple to white, which is exactly what Mendel found. And this is the F2 generation that was created. So if we read off the genotype for each of these, we can say that this one's homozygous, this one's heterozygous, this one's heterozygous, and this one's homozygous. And then finally, the last law that he came up with was the law of independent assortment. This is where each pair of alleles segregate independently of each other. So it was believed that when you have two different genes, so in this case we have the gene for seed color and we have the gene for texture of seed. So we have seed colors yellow and green, and then we have the texture of smooth and rough up here. So what was believed is that these were connected, which we can see is wrong. Because when Mendel ended up performing his experiment, he ended up getting variations. He didn't end up getting a smooth seed yellow with a rough seed green. He ended up getting these different variations. And we'll eventually get into this type of Punnett square here, which is known as a dihybrid. So I hope you learned. I hope this helped. Have a great day.